Well, I've got huge pleasure this morning to introduce Pierre Montay-Gruel. Anyone who knows about Flash will have seen the wonderful papers he's been producing in this area, and he's going to talk this morning about biology of flash radiotherapy in vivo. Pierre, over to you.
Good morning, Karen. Good morning, Mike. Good morning, everyone. I'm going to share my screen now. Please let me know, Karen, if you can see it.
I can see it.
Okay, perfect. Good morning, everyone. Honored to be invited to give this talk about the biology of flash radiation therapy in vivo today. And first, I would like to give a huge shout out to the organizers of this FRPT meeting. They gave a lot of time and energy in organizing this meeting, and I'm sure it's going to be very successful. So the subtitle of my talk today is "What does Flash do?" Because I think it's probably the hottest question in the field at the moment. I'll start by giving a definition of flash radiation therapy, and it's not that easy to give a definition of flash. We know that it's a radiation therapy technique that is delivered at extremely fast, at ultra-high dose rate. Read it. But what does that really mean? Is it only fast, and how fast? Is there a threshold in time? Are there other beam parameters, especially, for example, the energy that are involved? And do we need to meet those beam parameters to observe the effect? On the other hand, we know that flash does not induce classical radiation-induced toxicity patterns on the normal tissue, and at the same time has a very good anti-tumor efficacy compared to routinely used radiation therapy techniques or conventional dose rate radiation therapy. And that's what we call the flash effect. But is there a threshold in toxicity on this normal tissue that can be accepted? And does it concern all normal tissues and all tumors? Also, I would like to emphasize that this flash effect is an in vivo observation. And Marie-Catherine already said it in her plenary lecture earlier. And that's very important to say. So if I had to come with a definition of flash radiation therapy, I would say that it's a radiation therapy technique that is delivered at ultra-high dose rates with specific beam parameters, and which is able to treat tumors without inducing drastic toxicities on the surrounding normal tissues. That's a very careful definition, and I'm sure it is going to be improved, but so far that's the best one I could come up with. And what is very interesting in this FRPT meeting, is that you will have dedicated sessions for every single part of this definition: the beam parameters, the physics, the chemistry, the biology, tumor, normal tissue, mechanisms, etc., etc. So that's very, very exciting. We will start by the normal tissue. Always been called flash. And decades ago, between 1960-ish and 1982, many teams in the world started investigating ultra-high dose rate radiations. And the first experiments that have been done in vivo were performed on skin models in rodents. And Inada and collaborators in 1990 already showed that when they increased the dose rate above 2 times 10 to the 5th grade per second instantaneous dose rates, they observed that the toxicity on the skin of mice was reduced compared to dose rates under this threshold. A bit later, 2 years later, Hendry and collaborators showed that the necrosis dose 50 on the tails of rats was largely and significantly increased when the dose rate in the pulse was above 10 to the 5th grade per second. So almost like 40 years ago, it was showed that dose rate matters. And between 1982, after that, almost nothing until 2014 and this paper that you all know from Vincent Favodon. So what's new in 2014? Vincent Favodon in his paper and the rest of the team showed that flash irradiation for a similar dose compared to conventional irradiation did not induce lung toxicity, but at the same time on a lung orthotopic model, there was a very good anti-tumor effect of flash and they were able to increase the survival of tumor-bearing animals. So for the first time, it was showed that there was no normal tissue toxicity, but at the same time, a very good anti-tumor effect, meaning that this new radiation therapy technique could increase the therapeutic index of radiation therapy. And that was coined the FLASH effect. And after this study, after 2014, a large number of studies investigated the effect of FLASH radiation therapy on normal tissues, especially on the brain. Many experiments have been performed on the brain showing that FLASH radiation therapy was protecting the brain from radiation-induced normal toxicity. Mouse skin, bone, muscle, mouse lung, mouse gut, mouse hematopoiesis. Other studies have focused on other animal models, such as zebrafish embryos, but also bigger mammals like minute pig skin. More recently, investigations on canine skin, so veterinary clinical trials have been performed, veterinary clinical trials on cats, and a first human compassionate treatment was performed in Lausanne in 2019. And the common thing among all those studies is that FLASH radiation therapy delivered with electrons, protons, or photons was able to induce less normal tissue toxicity than conventional irradiation. But which parameters shape normal tissue toxicity? Well, in radiation biology, we know that there are 5 principal parameters that shape normal tissue toxicity: the total dose, irradiated volume, irradiation schedule, single dose or fractionated, the organ obviously is important, and individual variations. When we compare conventional to FLASH radiation therapy in the study, we usually do it at isodose. We use the same irradiated volumes, either whole organ or local irradiation. We compare similar irradiation schedules. Obviously, if we want to compare both techniques, we use the same organ. And in preclinical setups, we use genetically identical animals with a large sample size to avoid individual variations. So according to the handbooks, we should expect similar normal tissue toxicity between conventional and FLASH radiation therapy. That's not the case. So how does FLASH induce less normal tissue toxicity? And probably the beam on time is very important in this equation. So I have here a very nice figure that was just published by the group in Lausanne by Uda Kassem and Emric Almeida. And if we look at this figure, you can see here all the events that happen after the exposure of a biological tissue to ionizing radiation. So first, the physical, physiochemical, and chemical steps, biochemical steps, and biological steps. And here, we want to look at what happens at the biology level. So look at different at the organ.
Yeah, we're losing sound again. So could you try again? Could you start this slide again, if you don't mind? Thank you. We saw that one. We've seen that slide. So it's this. It's from the start of this one. Thank you.
Molecular points, given organ, and to see what FLASH was really doing. And I'm sorry if I'm not showing your favorite study, but it's impossible for me to go through all of them. So I picked just one each time. When people looked at DNA damage, it was observed that FLASH induced less DNA damage, especially gamma H2AX and 53BP1 foci in irradiated tissues. Same for cell death. It was observed by many groups that cell death was less induced by FLASH radiation therapy than conventional, especially apoptosis and senescence. For stem cell progenitors and progenitors depletion, same thing. So we published in 2017, we really showed that there was less, a better pool preservation and proliferation of progenitor cells after FLASH irradiation compared to conventional. Vascular damage, same thing. A very good preservation of the vascular system after endothelial cells after FLASH irradiation compared to conventional. Same for inflammatory response. with almost no inflammatory response after flash compared to conventional. And for the organ function alteration, there is a very good protection. So actually, flash radiation therapy does not do anything. And what you can read in the papers is the flash radiation therapy group is similar to the non-irradiation control group, or flash radiation therapy does not induce X or Y endpoint. So we are at the situation where we have a control non-irradiated group, a conventional group that is different from the control, and a flash group that is similar to the control, which is not very fun for us because we cannot understand how flash works if we don't see anything. So what does flash do really? And that's when we really have to look for a situation where we have a control group, a conventional group, and a flash group that is different from both control and conventional groups. And some studies have managed to find something like that. And that's what we've done in 2020 with Munjal Acharya. And we looked at astrogliosis after flash irradiation in the whole brain with a single dose of 10 gray. And that's what's shown before, but we observed that flash did not in the mouse brain after irradiation compared to conventional irradiation here in red. And we know that after irradiation, we have an activation of the complement cascade, especially an expression of the C1q protein that leads to the production of anaphylatoxin C3A and C5A, leading to chronic inflammation and astrogliosis. So we decided to look at the expression of C1q and observed that actually in the flash situation, we had a large and of C1q expression in the astrocytes. And if we looked at the anaphylatoxin, there we saw that the levels of anaphylatoxin C3 expressed in the astrocytes was similar between conventional and flash. So we really had a discrepancy in the cascade where we had three situations. In the control, no astrogliosis, no expression of C1q, no expression of C3. In the conventional group, we had astrogliosis, low expression of C1q, very high expression of C3. And in the flash, no astrogliosis, very high expression of C1q, very high expression of C3. So we really had something different in each group. If we look at this cascade again, we know that between the expression of the anaphylatoxin and the chronic inflammation, the intervention of TLR4 expression that acts on this cascade through PI3K pathway. So we decided to have a look at this TLR4 expression and observed that actually after conventional irradiation, we have a large increase in TLR4 activation, but after flash, there was none. So we really found probably one pathway that was differentially activated after both non-irradiation, conventional, or flash irradiation. Here, I want to emphasize that flash irradiation is really disrupting our knowledge of radiation-induced normal toxicity. We have two conditions, two techniques, conventional and flash. We use it as those same volume, same machine, same fractionation schedule, identical animals, and a large number of them to avoid individual variation. And we have a different normal tissue response. It means really that other radiation delivery parameters shape the normal tissue toxicity. And I will emphasize that it's probably the time, the beam on time is very important. So we have to understand the flash effect, we really have to choose the models wisely, probably to adapt them to this new situation, develop new ones. And so we can answer to the question, what does flash do exactly? And not only say flash doesn't do, etc. So I showed you that flash and conventional induce different toxicity on the normal tissue at the biological steps. But what if this difference was induced by differential activation of earlier pathways in vivo, meaning the physiochemical and chemical steps that could shape this difference between flash and conventional? So in 2019, we looked at the influence of oxygen in the context of flash irradiation and showed that when we were increasing the oxygen tension in the brain, we had a loss of this flash effect and a loss of normal tissue protection. Same on zebrafish embryos using antioxidant molecules. So we said that probably there is a consumption of the oxygen by this very quick irradiation, this very fast flash irradiation that protects the normal tissue. And between 2019 and 2021, no experiments had been performed in vivo to prove or right or prove us wrong for this hypothesis. Until very recently, and Cao and collaborators showed that actually there was a consumption of oxygen induced by flash more than conventional irradiation. Even more interesting that it was higher, this oxygen consumption was higher in normal tissues than tumors. Of this study was that the magnitude of this change is much smaller than would be required to achieve global tissue hypoxia in normal tissues that are well oxygenated. So the hypothesis was not completely wrong, but though it's not enough to explain the flash effect. So we still have to go further in vivo to understand how the early events, chemical and physical events, can shape the biological response. What about the tumors? Many models have been used to investigate the anti-tumor effect of flash irradiation. There were subcutaneous models, there were orthotopic xenografts, including human xenografts on mice, there were syngeneic orthotopic models, and there were spontaneous tumors like the cat, the dogs, or the patients. And all of them showed an overall isoefficacy of flash and conventional irradiation on the tumors. And all of them had their advantages and disadvantages in terms of potential clinical application or mechanisms investigation. If we look at all the orthotopic and spontaneous models that were used, we have different types of tumors that were both with flash electrons and flash protons. Murine glioblastoma on the brain was investigated, murine lung carcinoma, murine ovarian cancer, murine lymphoma, murine head and neck cancer, feline squamous cell carcinoma, human skin lymphoma, and canine cancers. I would like to draw your attention that on the fact that way less studies have investigated orthotopic or spontaneous tumors than normal tissue toxicity. So flash radiation therapy does the job on the tumor. And we can read in publication that flash radiation therapy group is similar to the conventional radiation therapy group, or that flash radiation therapy is efficacious to increase tumor growth delay. So we have the situation where we have a control non-irradiated group, a conventional group, and once again, flash looks like conventional. That is not very helpful. We would like to understand how flash works on the tumor as well. So are there situations where flash radiation therapy is different from the control? Can flash be better or worse than conventional radiation therapy? And we would like to see a situation where the control is different from conventional irradiation and is different from flash again. So one study was published very recently by the group in Stanford, and that was performed on the tumor microenvironment. We know that radiation therapy is very important on the tumor microenvironment, and especially essential in the context of immunotherapy. So there was a study that showed that flash does not induce collapse of the vascular compartments in the tumor, and that was linked to a better perfusion and thus an increase in the infiltration of tumor infiltrating lymphocytes in the tumors. And we had different situations. Unirradiated controls where TILs were less present in the tumors, conventional induced better infiltration of lymphocytes, and flash really induced a way better infiltration of lymphocytes in the tumor. So here we might have something that is better with flash than with conventional irradiation. On the other side, another recent study published by Shabi and collaborators was focusing on T-cell acute lymphoblastic leukemia. And they used 3 different models of T-cell acute lymphoblastic leukemia injected in mice. And they showed that for 2 of them, Flash and conventional were at least similar, with, for one model, maybe a slightly better effect of flash compared to conventional irradiation. But on the third model, unfortunately, it was observed that after conventional irradiation, all mice survived, whereas after flash irradiation, all mice died as the controls, meaning that for this situation, flash must be worse than conventional irradiation, or at least not as good as conventional irradiation, or maybe tumor. So there are conditions for which flash might not work. Flash radiation therapy is efficacious against tumors, but we know that it's good for high single doses, hypofractionated regimen, small volumes, and tumor growth delay. And there is a real lack of studies to investigate fractionated and hyperfractionated regimen on other models than glioblastoma, the volume effect, tumor heterogeneity, treatment combinations with other techniques, treatment planning, and local control. Also, there is this mechanism on the tumor that we don't know exactly how it works yet on the tumor and that we need to investigate. And I want to emphasize that tumor control is just as important, even if not more, than normal tissue preservation. So we really have to investigate that to ensure a safe and efficacious clinical translation of flash radiation therapy. And I want to finish my talk by saying that the flash effect opens a huge potential for cancer research. Because after flash irradiation, we have a protection of the normal cells, but we also have of the tumor cells. So flash radiation therapy actually investigates, or actually has a differential effect on both types of cells. And there is probably an intrinsic biological difference between normal cells and tumoral cells in vivo that can be outlined by the flash effect. So we could use flash not to treat the tumors only, or to go towards a clinical application, but really to investigate this intrinsic difference between normal cells and tumor cells on a fundamental research plan. So what are the take-home messages? There's still a lot to do before understanding the flash effect, mechanisms behind the absence of normal tissue toxicity and behind the anti-tumor effect. Answer to the question, what does flash do? And not say only flash does not do, etc. Ultra-high dose rate irradiation is disrupting our knowledge of biological tissue interaction with ionizing radiation. We have to accept it. We should not have expectations on the results we observe, and we should brainstorm to find new models, new hypotheses, and solutions to investigate that. And also, flash does not only have a clinical future, but it's also a very powerful tool for fundamental research. So I would like to end my talk now by showing you my very small but very nice team here in Antwerp, Belgium. And why don't you join us? We are recruiting PhD students and lab assistants at the moment. So don't hesitate to contact me or just contact us if you want to participate to this adventure. Thank you very much. I hope not running too much over time, and I hope you could hear me properly.
Pierre, thank you so much. And brilliant talk. Really, really enjoyed it. I'm going to hand over to Mike, who's going to handle the questions. So Mike, over to you.
Yes, thank you, Pierre. As you might anticipate after such an exciting talk, there are a number of questions from the audience. If I could start with the first one from Guillaume. It's because of the big amount of parameters and the hypothesis, including beam on time, what needs to be published from this type of work? Is it only the successful work or all works, including the non-successful ones, so that we can compare the parameters?
Great question. I think we should really publish everything, the positive and the negative data. It does not only concern flash. Negative data are very important in the whole research fields, but in the flash, in this very new research field, we must understand when it works, when it doesn't work, to answer the question, why does it work and what does flash do? So don't hesitate to show your negative data. It could be combined to positive data. It could be only negative data. Just be careful about how you present your negative data, not to say flash is not a thing or et cetera, but it's very important for the field to compile all these results.
Thank you. So we have a question from John Ely. Could you talk about the dose levels that you found where flash did not appear to spare the rodent brain?
Dose rate or dose?
The dose levels. I suppose dose and dose rate.
The dose levels. So what I have observed in my hands is that when we reach a dose above 14 gray single dose, we are losing the protection on the normal brain. But that was with our setup in Lausanne, with our animals. And I know that other teams, and it's been published by the group in Stanford, for example, were able to go higher in dose with single doses, and they still observed the flash effect. So it's very difficult to answer this question because I think it will really depend on the setup of your irradiations, how you irradiate the animals, which organs are in the field. If you have the mouth, for example, in the field, it will be different. So it's difficult. There is no lower threshold, though. We know that conventional irradiation induced drastic cognitive toxicities even at lower doses. So we investigated as well lower doses and saw that flash radiation was protecting the brain from normal tissue toxicity.
Okay, thank you for that. So another question from Luke Dykes. Would you be able to comment on any new facilities or machines that are needed to complete more systematic or targeted studies of the flash effect?
I didn't hear the first part of the question. That was cut.
Apologies. Would you be able to comment on any new facilities or machines that are needed to complete more systematic or targeted studies of the flash effect?
If I understand the question is, do we need new type of facilities to investigate the flash effect? I think any kind of machine or facility that is able to deliver ultra high dose rates, it could be any kind of beam types, is interesting. We really want to see if flash is linked to only the dose rates or other beam parameters. So I guess if you have a machine that is able to reach very high, ultra high dose rates, it is worth it to investigate if you can achieve the flash effect. That would be my answer. There is no real limit.
Okay, do we have time for one more question, Karen?
I think we can probably just squeeze one more in.
Okay, perhaps I'll be a little bit selfish and ask my own question then. So Pierre, our understanding of conventional radiotherapy is quite well characterized by the linear quadratic model. Do you think that the normal tissue sparing that goes with flash will follow a similar response in where we understand that a high alpha beta ratio or a low alpha beta ratio characterizes the response of different organs?
My opinion at the moment is probably that the tools we are using to characterize conventional dose rates irradiation might not all fit to flash radiation therapy. I don't know if the linear quadratic model alpha beta ratio will fit with flash, the flash effect, but we will probably at some point have to gather around the table and look at this alpha beta ratio, linear quadratic model, RBE, oxygen enhancement ratio, absorbed dose, and look at if those parameters actually matter or can fit into a flash irradiation model or not. So for the moment, I have no real answer to that. I think it needs to really be investigated, but those questions are very important. And I said it, flash really disrupts our knowledge on normal tissue toxicity. And just like, let's dive into it and let's try to understand if the tools we're using are outdated for flash or can be adapted.
Thank you, Pierre. Thank you for a brilliant talk, and thank you to the audience for listening. Andrea, you have asked a question, and we'll bring that question up on on the in vitro side later on today. So please, we will answer it, I promise. So thanks again, Pierre. Huge thank you. And we've got another session about to start. So we will see.
Thank you, Karen.
Take care.
I will share the mechanism. So see you in the mechanism session. Bye-bye.
Mechanisms. Take care. Thanks again, Pierre. Excellent talk.
Bye.
Loads of comments about excellent talk. Thank you.
Thank you. Bye-bye.
Bye.